Hi, this is Yogathisan from University of Illinois. I'm going to talk to you about my paper titled EEG-GRAPH. This is a joint work between the Mayo Clinic and the University of Illinois at Urbana-Champaign. A few years ago, former President Barack Obama announced the Brain Initiative, whose primary goal is to map the entire human brain. Many organizations, including NIH, DARPA, and the Kavli Foundation, are active members of this initiative. Through Mayo Clinic, we contribute to some of the studies under this initiative. In many of these studies, a common way of collecting data from the human brain is using electroencephalograms or EEG. Here I am showing an EEG recording collected using an electrode array directly implanted on the brain. In this particular case, we are interested in identifying spiking events like these, which we refer to as abnormal events. The exact pattern of these events may vary depending on the research problem, and identifying these events is paramount for arriving at meaningful conclusions. Manual identification is cumbersome and it does not scale very well. Therefore, researchers are interested in developing tools that can detect these type of events automatically. Most of the existing works utilize the spectral characteristics of these events to detect them. But these methods suffer greatly from signal artifacts. In our paper, we present a graphical model-based approach that uses some additional attributes of these events to more accurately detect them. We observe that adjacent electrodes in the brain show similar patterns in emitting abnormal events. In addition, some electrodes like this may emit these events repeatedly. So we utilize the spectral characteristics of the event, its spatial patterns, and its repetitiveness to identify whether a particular window of EEG has an event of interest. As shown in this diagram, the presence of an abnormal event depends on the observation, which is basically the spectral characteristics of the event, and its previous states, and also on the adjacent electrodes. Due to the flexible platform provided by graphical models in general, we have adopted to use a factor graph based paradigm to model this. Factor graphs are specific type of graphical models. And we use random variables to represent the different entities in the graph and factor functions to represent the relationships between them. To show the feasibility of the model, we tested it for the application of seizure onset zone localization for patients with drug-resistant epilepsy. We defined the factor functions based on expert knowledge for the specific problem, and based on our definitions, we show that a graph cut based approach can be utilized to efficiently perform inference on this graph. Our results indicate that our model performs significantly better than currently existing methods with very minimal computational effort. Now to summarize, we developed a model based on probabilistic graphs to encode observational, spatial, and temporal relationships in brain activity and address a very important research gap in current brain research. Thank you.